Forbasis is a Swiss company with the headquarters in Lugano and production facility and research and development labs in Italy, in Pavia, which is near Milan. And Forbasis is certified for the development and production of medical devices. And so the main mission of Forbasis is to make available to clinical labs medical devices in order to improve precision medicine. We have in particular two technologies for the development of our kit: the family of panels kit, which are amplicon-based kit, and the family of Pro kits, which are capture-based kits. In particular, today we will speak about COVID panel, which is from the family of panel kits, so amplicon-based kit, and is based on the version 4.1 of the Arctic amplicons, and for the entire sequencing of the whole genome of the SARS-CoV-2. And as every panel of our kits is available both for Illumina and Ion Torrent platforms. Let me give the shine to Martina, our NGS application specialist, in order to present in detail the protocol of the panel.
Hello, everyone. In the current slide, you can see an overview of the COVID panel protocol. As said before by Valentina, the kit must be used on both Illumina and Ion Torrent sequencer. The starting material is cDNA that could be obtained with any commercial kit for retrotranscription. And for each sample, the starting input must be 30 nanograms. The first step of the protocol is enrichment PCR for the target amplification. And after that, you have to quantify your library, because for the next step, you need 100 nanograms of each library. The second PCR is for indexes or barcode segregation. And at the end of the PCR, you must purify your libraries and then proceed with the quantification of each single library for the creation of a smaller pool. Once you have obtained the final library, you can proceed with the dilution and the loading on your sequencer. In the next slide, there is an overview of the benefits of COVID panel kit. Is a very quick protocol with hands-on time less than an hour and a half, and is validated for the detection of all known variants and optimized for Omicron 1. The entire genome of SARS-CoV-2 is covered, and the kit is offered in a bundle solution with a dedicated analysis software. In this slide, I would like to introduce you to our latest news, that is COVID panel fast. This version of the protocol is only for research use for now, but with this protocol, the hands-on time is further reduced under 1 hour. The starting input must be 30 nanograms, like the standard protocol, and after the first PCR is not required quantification, because for the second PCR we have to use a fixed volume of each library. And after the indexing or barcoding PCR, we directly pull together all the samples with a fixed volume. And after that, we perform the purification in a single tube. At the end of the purification, you have only to quantify the library and proceed with the dilution and loading on the sequencer. The next slide is to make your attention about the length of amplicon. In fact, the amplicon of COVID panel kit is about 400 base pair. And this means that is required a sequencing chemistry of at least 500 cycles. Another important information is that if you need, we could provide a positive control cDNA that is a control ready to use for the protocol. And as said before, the starting material is about 30 nanograms for each sample. And now Valentina, okay, sorry.
Regarding data analysis, we developed our data analysis on our proprietary platform. The name of the platform is Forever. And here you can find the link to our platform and the first overview. The workflow is very simple. You can upload your FASTQ file on after login. You can upload your FASTQ file in the platform, and then after 15 to 20 minutes, you will have results, and you will receive an email with the advice of these results available. Results for COVID panel are coverage metrics, WCF annotated filters, filtered and raw, with the variants identified, a BAM file for the visualization on IGV, a consensus sequence in FASTA format, which you can upload on GSAID or Netsclade or Pangolin as an example, and a file with results directly calculated by the pipeline in Netsclade and Pangolin, and a PDF report in which you can find a summary of the coverage metrics, the variance list, Pangolin and Netsclade results, and a visualization of the spike protein in 3D with the variants identified. COVID panel is available in two formats, for 96 or 384 samples, of course, with our indexes, both for Illumina and Ion Torrent sequencer. Thank you for attending this brief overview. And now, please, if you have questions, we are here to reply to you. There's a first question asking which primers are used. The primers are more or less similar to the primers in the Arctic project version 4.1 in order to cover also the Omicron variants and Delta and Omicron variants. And in fact, we tested and validated the panel on both Delta and Omicron in synthetic samples. There's another question about the concentration, and I'm not sure of what do you mean with the low concentration, but yes, we validated the panel in low concentration. But the perfect usage in the IVD for the IVD kit is in the clinical concentration calculated in RT-PCR, so between 15 and 30 CT.
Yes, the kit could run on Ion Chef automation, and it cannot run on ISEC because of the chemistry. We need a chemistry of minimum 500 cycles.
And so it's not compatible with Minisec cause of the needs for a minimum of 500 cycles. Yes, the kit is ready to order. And the delivery time is 1 week in this moment, both for the 96 and the 384 format of the kit. And the problem of the chemistry is related to the complete coverage of every amplicon. We tested also in ISEC, but only for research. If you really want a complete and deep coverage, we suggest, in particular for CAVD, a chemistry of 500 cycles. Next question is, what consumables are required? I think Martina can reply.
Yes, is required a kit for cDNA synthesis, so for retrotranscription, and ethanol, and beads for purification. We suggest impure XP beads for the purification of the library.
We hope to have replied to all your questions. If you need more questions, if you need more info, you can, of course, write to our email info@4bases.ch or info@4bases.it. And if you have other questions, please, or I think we can stop this session. Thank you all to have attended this meeting, and we hope to hear from you soon. Bye to everyone.
Thank you.